Hey Team Bio, I think the first thing you'll notice when you take a look at this image is that I've actually drawn a lot more in here than I gave you on your original note. And the reason for that is I handed out the note before I had recorded the screencast, and I was drawing all of the components and it was just taking way too long. It was turning into a 20-minute screencast. I didn't want that to happen. So instead, I filled some of it in, and yeah, so I apologize for that. You can find this blank note fully fleshed out like it is now on our the Schoology page along with this assignment, and you can reprint it if you are annoyed at the fact that you don't have all of the drawings that are in here. So again, apologies, but hopefully the screencast won't be quite as long. Okay, so this screencast is going to be about anaerobic cellular respiration. Now, I introduced you to this flowchart through cellular respiration yesterday, and you know that both types of cellular respiration, aerobic and anaerobic, both begin with glycolysis. Anaerobic happens in the absence of oxygen, and aerobic happens in the presence of oxygen. Organisms, there are two types of anaerobic cellular respiration: alcoholic fermentation and lactic acid fermentation. We are lactic acid fermenters, and yeast are alcoholic fermenters. We are actually creating alcohol over in classroom 215 right now in the inverted test tube lab. So let's get right to it. I guess the last thing I want to say is, even though I'm introducing glycolysis in this anaerobic cellular respiration screencast, know that glycolysis is also the first step of aerobic. Okay, so what is glycolysis? Glyco meaning relating to sugar, and lysis meaning to rupture or to split. Glycolysis is just the splitting of glucose. Now, you know the chemical formula for glucose. It's C6H12O6. Know that I am going to represent glucose starting out here as just carbons. I'm only going to show the carbons moving through the pathway, but know there are a bunch of functional groups hanging off of these in the form of hydrogen and oxygens. But I'm just going to simplify it and not draw out the whole structure. So I'm only going to represent the carbons moving through here. Okay, so we start out with glucose. Glucose enters glycolysis, and the first phase of glycolysis is an investment phase. And the investment that we're making is in the form of ATP. So we have to spend a little ATP in order to get out the 2 ATP that are created in glycolysis at the end on net. So what happens here is that glucose enters glycolysis, and we drop off 2 phosphate groups on either end of the molecule here and here. And we charge up the glucose. We make it more energized than it already is. And spending these 2 ATP leaves us with 2 ADP. The next phase is called the redox phase. A redox reaction is a reduction-oxidation reaction. There's a mnemonic that can help you remember what these reactions do. Leo goes Ger. The loss of electrons is oxidation, and the gain of electrons is reduction. So whenever we have a reduction oxidation reaction, something is reduced and something is oxidized. Something loses electrons and something gains those electrons. And in our situation, we have this 6-carbon compound getting split up into 2 3-carbon compounds. There were these phosphate groups that travel along with it here and here, and we actually get 2 more phosphates coming in, charging up either ends of these 3-carbon compounds. So now we have 2 3-carbon compounds that have phosphates on either side. And then this is coupled with a redox reaction where electrons, high energy electrons, are stripped from this 3-carbon compound, which is called G3P. And the stripping of these electrons is passed off to a molecule called NAD+. So just like we saw in photosynthesis, NADPH was our high energy electron carrier. In cellular respiration, we have another similar compound called NAD+, which becomes NADH after it picks up some electrons and hydrogen from G3P. So because NADH picked up electrons, it gained electrons. NADH is reduced and G3P is oxidized. We're going to take these electrons, if we have the presence of oxygen, over to electron transport phosphorylation, but we'll learn today that in fermentation, we are going to recycle them in the fermentation pathway. So this is important. NADH is picking up high energy electrons from G3P. And then the last phase of cellular respiration is the payoff phase. And this is where we swoop in here with 4 ADP and we pick up all of the phosphate groups that were bound to the edges of the G3P. So we create 4 ATP at the end here, and we're left with a 3-carbon compound called pyruvate. So we entered with 1 6-carbon compound called glucose, and we ended up with 2 3-carbon compounds called pyruvate. We spent 2 ATP, and we got out 4 ATP and 2 NADH. So our total accounting is on net. We have 2 ATP and 2 NADH. In the presence of oxygen, we are going to pass these electrons down an electron transport chain in electron transport phosphorylation. And that's going to make a bunch of ATP for us. But in the absence of oxygen, we have to do something in order to free up this NADH and return it back to NAD+, so we can keep running this pathway, glycolysis. And that is what the fermentation pathways are all about. Now, it maybe doesn't make a lot of sense to introduce them before we talk about electron transport phosphorylation, but what the heck, doing it anyway. I guess we're talking about it because we're doing fermentation right now in lab. Okay, so the whole point of the fermentation pathways is to free up this NADH, return it to NAD+, so that we can keep doing glycolysis and keep getting this 2 measly ATP per glucose molecule in glycolysis. So pyruvate, so we're going to have another redox reaction where now, instead of being reduced, NADH is oxidized. It's going to pass off its high energy electrons and make more NAD+, so glycolysis can keep happening. Okay, so pyruvate enters either alcoholic fermentation or lactic acid fermentation. These 2 3-carbon compounds are both pyruvate. And in alcoholic fermentation, water is involved in the reaction. It's going to come in and help split the carbon off of this 3-carbon compound. And this is going to be CO2. Created, and we're going to see evidence of this CO2 in the form of a bubble in our inverted test tube lab. So that hydrolysis reaction is going to lead to a 2-carbon compound called acetaldehyde. Which this is part of another reduction oxidation reaction where NADH comes and drops off electrons onto acetaldehyde, and NAD plus is created. This NAD plus can go back to glycolysis and keep running glycolysis. The end result is ethanol. Informally known as alcohol. This other pathway, the lactic acid fermentation pathway, also starts with pyruvate. See you later, Chrissy. But instead of making CO2, NADH comes in directly, drops off electrons, and makes NAD plus, and the end result is lactic acid, which is a 3-carbon compound. So, alcoholic fermentation is going to lead to the production of CO2, whereas lactic acid fermentation will not. But they both create NAD+, meaning glycolysis can keep running. Okay, that's all. Slightly boring screencast, but hopefully it will help bring some clarity to what is happening in the inverted test tube lab. Okay, see y'all tomorrow.